This video will talk about classical genetics. These are some of the techniques that were invented before the molecular biology revolution, and we're going to focus on things that are still useful. The first one of these is selection. This is where you set up media conditions in such a way that only mutants of interest are able to grow. For example, you take a bacterial culture, you might mutagenize it in some way with chemicals or ionizing radiation or whatever, and then you put it on a plate. And say you put about 0.1 ml, which is about 10 to the 8 cells on this plate. This plate contains the antibiotic rifampicin, and the only things that can grow on this plate are organisms that are resistant to the drug rifampicin, and you'll see them grow up. Now, I want to take a little tangent and talk about what actually has happened there, because this is a common misconception that students have. Did the mutations arise because of the presence of the antibiotic? In other words, they induced mutations, or did these mutations spontaneously arise while the culture was being grown up in the test tube and the antibiotic just selects for them? This is a classic question that they had back in the middle of the 20th century, and it was answered by an ingenious experiment by Salvador Lori and Max Delbruck. In this experiment, they look at resistance of E. coli to phage P1. Normally, if you plate out 10 to the 8th of coli on a plate that has been exposed to phage P1, most of them will die, but some of them will still grow up and be fine. If the mutation occurs before, you would expect a wide variation due to siblings or out that came up during growth, and I'll explain that more in a second. If mutations occur during the plating, when they're on the plate, they're under the selective pressure of P1, you would expect the same number. So let's dig into that a little bit more so you understand. We're going to do two experiments. In the control, we're going to grow up a single culture. We're going to separate it in 10 separate portions, and then we're going to plate 10 plates from each of those portions. So we're going to end up with 100 plates. In the test, we're going to grow up 10 cultures independently, and we're going to plate 10 plates from each of those cultures. Now, what do we think is going to be going on there? If a mutation occurs before selective pressure is applied, during the growth of the culture, before it's put on the plate, each change to resistance will not be independent. To understand why, think about the time the mutation occurs. If the mutation occurs early when the culture is growing up, shown here on this second panel here, right? The mutant strain has a chance to multiply, and there will be many bacteria in the culture that are phage resistant. If the mutation occurs at the end, near stationary phase, only a few bacteria, shown here in the fourth panel on the bottom, or maybe no mutation at all, only a few bacteria will be phage resistant. Thus, the number of mutants is dependent upon the time of mutation. In contrast, if the mutation is induced by the selective pressure, each change will be independent. As you can see here, no mutation is occurring during the outgrowth. They're occurring once they sit on the plate, and they're all independent from one another. So an equal portion is spread on each plate, and each cell in the population has an equal chance of mutating to phage resistance. Thus, you should observe about the same number of mutants on each plate. If mutation events are independent, and thus the induced panel A here, the induced mutation is what we're talking about, then they will follow what is called a Poisson distribution. And one of the characteristics of a Poisson distribution is that the mean will roughly equal the variance. However, if mutation is occurring before selective pressure is applied, the occurrence of mutation is not independent. It is dependent upon the time of the mutation. It will not follow a Poisson distribution, and the mean will not equal the variance. So in our control, since we're using a population of cells that came from the same culture, you should see the same number of mutants on each plate, right? That one culture will grow up. Let's say there were 2 mutants in it, right? When you split it into 10, you're still going to see 2 mutants per 0.1 milli, you plate on the plate, you plate out the 10 plates, you're all going to see about the same number. However, for the test, remember it's 10 different cultures. A mutation to phage resistance can happen at any time because they're grown up separately, and their number of mutants is going to be dependent upon when the mutation occurs. If the mutation occurred in the initial culture. So we can test this out using the mutate program. I've now started up the mutate program, and what you're seeing is the DNA sequence, at least the first part, for the ferrochrome protein, which is outer membrane protein, which is actually the receptor for phage P1. If you mutate this such that it's no longer there or it's not the same shape, P1 can never bind, and then you are phage resistant. So we're going to do that. We're going to change this base 13, and we're going to change this from an A to a T, and what that does is it causes a stop codon. All right, now we're going to do the experiment. It's called a fluctuation test, and experiment 1 is the control. And again, we're going to plate about 10 to the 8th bacteria, and the mutation rate we're going to keep the same in both because we assume it's the same. And then we're going to have 10 cultures, but again, remember they all come from the same. We're going to have 10 cultures that all were split off from the same culture, and we're going to make 10 replicates. In experiment 2, same number of bacteria, same mutation rate. This time we're going to grow up 10 separate cultures, and then we're going to plate them out. So again, experiment 1 is the control. We grow up 1 culture, split it into 10, and plate it. And experiment 2 is the test, where we grow up 10 separate cultures. All right, what do you predict is going to happen? Do you think that mutations are induced by the selective pressure, and therefore the mean and variance is going to be equal in the control and the experimental? Or do you think mutations occur before? They're spontaneous and the selection only selects for them. They're not occurring on the plate. Okay, make your guess now. All right, so let's run it. We run the experiment, and what we find is that in the control, as expected, it follows a Poisson distribution, and the mean and variance are equal. But in the test experiment, where we grow up 10 separate cultures, the mean is 8.8 and the variance is 1,351. So they're not equal. So we can run this many, many times, and you see in all cases, in no instance does the mean equal the variance. Therefore, what this means is that we're following the spontaneous mutation model, and mutations occur in populations before the selection, and the selective pressure just finds them. Okay, we're done with that now. Let's move on to another way of finding important things that you're interested in. So let's imagine that you want to find an enzyme activity. You want to see its increase or see if it's decreasing. Screens are another way of doing it. If you can't select for your mutation or your change, you will use a screen to find it. In screens, most cells grow and the desired mutant is detectable. You can detect it in some fashion. You use screens when there is no good selection, and there's a number of different types of screens, and we'll talk through them. Assays, you can assay an enzyme that you're interested in. You can use replica plating. You can use color indicating plates. So in an enzyme assay, let's imagine you're looking for the increase in the activity of an enzyme in a strain, so that you mutagenize this strain. You now have supposedly mutants in here that are actually making more of the enzyme, or the enzyme is more active. You then dilute it out and you plate them in plates so that you get thousands of isolated colonies. You pick each colony, you lyse it, and assay the enzyme. You keep track of where all these colonies are, and then the desired mutant that has the activity you're interested in, you can then find it on the plate because you remembered in these thousands of colonies where it was. Obviously, this is a very labor-intensive way of doing this, and you can probably only screen through a few thousand colonies or tens of thousands of colonies doing this. Unless, of course, you have an undergraduate that you can pay to do it, or you have a robot that can do it for you. Another thing that you can do is replica plating. Let's imagine you're looking for a mutant that can no longer grow on histidine. And histidine is an amino acid that an organism can synthesize as part of its biosynthetic pathway. So you grow up a master plate, and on this master plate you plate the cells, you mutagenize them, and then plate them out on a minimal plus histidine plate. You take that master plate and you press it onto velveteen, right? And velveteen is this fabric with little hairs in it, little threads that will pick up the colony, and it kind of makes an impression, an imprint of the colonies that are on the master plate. You then put it on a test plate, and in this case, the test plate is minimal media without any histidine. So many of the colonies are going to be able to grow up on this plate, but there is going to be one colony or maybe a couple colonies on a plate that is not. So this organism cannot synthesize histidine. It can do everything else it has to do, because remember, this master plate was minimal plus histidine. You take away the histidine and it can't grow. So this is now a histidine mutant, the mutant that you're looking for. Another example of what you can do is color indicator plates. In a color indicator plate, you have some media where the mutant that you're looking for will appear a different color, and these are very powerful screens. So again, same thing, you take a bacterial culture, you mutagenize it in some way, you then dilute it out, and then you plate it for isolated colonies and incubate. Your desired mutant will look differently. Imagine that you plate onto a McConkey's plate, and again, remember that lac minus colonies will appear differently than lac plus colonies. So you can just plate them all out. You can look through hundreds of plates and screen through thousands of strains, and it's very easy to very quickly find a different colored colony that signifies the mutant that you're looking for. So there's 3 examples of the kind of screens that you can do. You can do enzyme assays, you can do replica plating, or you can use color indicator plates. Okay, we'll finish with a clicker question here. A culture of E. coli is spread on a plate of LB plus rifampicin, an antibiotic. Colonies appear on the plate. This is a selection, screen, or neither.